for Dr. Dekkers. Oh, I'm sorry, Dr. here. Dr. Dekkers grew up in the Netherlands and received a bachelor's and a master's degree from Wageningen Agricultural University and a PhD from the University of Wisconsin in animal breeding. From 1989 to 1997, he was on faculty at the University of Guelph, working closely with the Canadian industry on genetic improvement of dairy cattle. He moved to Ohio State University in 1997, where he is currently a C.F. Curtis Distinguished Professor in the Department of Animal Science. Current research focuses on genetic basics of feed efficiency and health in pigs and poultry, and on the integration of quantitative genetics and genomics in breeding programs. So I will turn it over to now Dr. Dekkers, and we can get started.
Okay, thank you very much, and thanks for organizing this. Great to be back. So as Julia mentioned, some faculty here from 89 to 97. Really enjoyed our time in Guelph. Still have lots of friends here. Some of them are here. Former collaborators, well, still collaborators, Laurie and Bonnie and Julie and industry, Gary and Flavio. Well, you weren't here when I was here, right? Did you start already? You were a student. That's right. Yeah. That's right. So, and then some online. Filippo was a student here when I was here. And so, yeah, lots of familiar. Good to be back, and good to see that the group is still going strong. So I was part of CGIL. I was actually looking for the group photo that we had that was hanging on the wall from group photo from that time, but it's not there anymore. No, not the lineage one. It was the group. Brian Kennedy and Ted Burnside, Charlie and John Gibson and Larry and from Jim Wilton. But yeah, very good time. I learned a lot during that time, but then we moved to the greener side of the border. Although now this looks like the greener side again, especially my wife, and maybe the fact that it's recorded, maybe you don't want to let us back in again. But she's from the U.S., but she would move back to Canada in a heartbeat. So, we're here. Actually, there's a wedding. We have a cousin, the daughter of a cousin of mine, is getting married in London tomorrow. So that brought us in the area, and so then made use of the opportunity to visit here and. Had a lunch and met with some students from Ray and some potential collaboration. So it's good to be back. So yeah, breeding for disease resilience in pigs. So yeah, when I moved from Guelph to Iowa, pigs are very big in Iowa, and so I also moved to work from dairy cattle on pigs. Filippo, don't sign off. This might still be interesting to you. So does anybody know who this might be? Yes, very good. So Charlie Smith was a faculty member here when I was here. He's originally from Scotland and was hired here in the CMAX chair as part of CGIL. And he passed away in '97, I think. Much too early, but he's always been my idol in terms of the work he did and his personality. Very kind. I think Bonnie, you interacted with him, right? Yeah. So this is a picture of Charlie when he was a PhD student at Iowa State University. So he grew up in Scotland, but at that time, Jay Lush, who is the father of animal breeding, had established himself at Iowa State and attracted a lot of graduate students from all over the world, and one of them being Charlie. And this is a very typical pose because he was always thinking. One of the things that Charlie was very well known for was mowed nucleus breeding programs and marker-assisted selection. And that was before or after his time as a PhD student, but you can just see the wheels turning. And he's looking at, this is Bircher Hall, is where the main administration of the university is. So yeah, so that's already a connection between CGIL and Iowa State. And nowadays, when people come and visit, so this is Charlie when when he was a faculty member here. Nowadays, we have people look up that rock. So it actually is a rock that has a, so it's facing Beecher Hall. Beecher used to be the president, and it actually is a plaque for Beecher, but we call it Charlie's Rock. And so this is Haja Kadamedin, who some of you may know. He was a PhD student with me and John Gibson when we were here. And if you look at this tree, it's still there. So, so you get, if you need inspiration, you go sit on Charlie's Rock. So, and then also in our library, we have all the theses from all the graduate students. Charlie did both a master's and a PhD at Iowa State, and he actually worked with Illinois Hazel, the selection index, but also the backfat probe. And Hazel was a faculty member there, but he was actually also a student of Jay Lush, who, as I mentioned, is the father of animal breeding. And one of the other students of both Lush and Hazel was Charles Henderson, mixed models. So a lot of there's a lot of history here, but a lot of that history connects back to the history at Iowa State. So it's quite a special place to have been here and now to be at Iowa State. I was going to show one other thing, if I can. That's not a team you guys would be cheering for, but this guy here is a nephew of ours, Ben Rortvedt. He's a backup catcher for the Dodgers. So we are kind of conflicted. When we were here, the Blue Jays won the World Series twice, and that's when I started. Well, those are the only games I actually have watched baseball that I've watched until when Ben started playing in the major leagues. So we're a little bit conflicted about who to cheer for. May the best team win, and that looks to be the Blue Jays nowadays. So right now. Okay, but now about disease. So infectious disease is the greatest challenge to pork production, not just pork production, but livestock production in general. Now there are pathogens where we have found, or people have found, that their resistance is controlled by a single gene. F18 E. coli is one example of that. But for most pathogens, resistance or susceptibility seems to be affected by more than one and possibly many genes. And of course, there has been a lot of progress that's been made to try to find genes. So we did a lot of work on PRRS in pigs in collaboration with Bob Rowland, who was a virologist at Kansas State, and Joan Lunny at USDA. I think Bonnie knows Joan very well. Where they did experimental infections, and this was with the industry consortium, the PRRS Host Genetics Consortium, which also included Genesis, where pigs from those companies that were part of this consortium were sent to Kansas State, and then were infected with a particular strain of the virus, and then we did GWAS, and we found quite a large effect. The GBP5, and we subsequently identified what we think is the causative mutation. This is also a gene that's involved in susceptibility or resistance to HIV in humans. We tested another genes. We found that same gene, and that's a gene or a marker that the industry has been using to some degree to select pigs that are more resistant. But it's still only explained about 10 to 15% of the differences in viremia. So it didn't make. They still got the virus, right? They still got infected. But of course, the PRRS edit, the gene editing. So Randy Prater's group at the University of Missouri, they knocked out the CD163 gene, which is the receptor that the virus uses to enter the macrophages. And in those same kind of trials at Bob Rowland's lab, they found that the knockout pigs didn't show any viremia compared to the wild type pigs. And in those experiments that I just showed, we evaluated, I think, up to 2,000 pigs that went through those challenges. Never a pig like this. They always got infected. So apparently that is a mutation, or that's a whatever that the effect of that gene, the mutation is not segregating in the population. And that's very successful, right? That's a big, and has been now been approved by USDA, FDA in the US. But there's many other pathogens, and even for PRRS, is it going to be resistant to all the strains or future strains? It's a virus that mutates very quickly, and then there's all these other pathogens. African swine fever is a big one. The Ray's group is working on that, and hopefully find a knockout or an edit that will make them resistant. But these viruses evolve, these pathogens evolve, and there's new pathogens that may come into the industry. So another approach is to focus on what we call polymicrobial disease resilience. So resilience of pigs to multiple diseases, and meaning the ability that if they get infected, they're still able to maintain performance. Okay? It doesn't mean that they don't get infected. They may get infected, but we want pigs that are able to clear the pathogen more quickly and keep growing, or go back to growing. So that's been the aim that we've been pursuing. So looking at natural genetic variation in resilience. And so just to illustrate this, if you have growth rate against disease pressure, pig A, both pigs, they have the same performance when there's no disease, but as you increase disease pressure, pig A is more resilient because its performance is less affected by an increase in disease pressure. Okay, and so that's one phenotype we can look for, or we can just look for the number of veterinary treatments that pigs require, or mortality on the disease, other measures of resilience that we've looked at. Now, the challenge of selecting for disease resilience by breeding companies is that the breeding program is structured like this, where there's a terminal sire line and typically two maternal purebred lines, Yorkshire and Landrace often. And they are kept by necessity in a high health environment, high biosecurity, very good management, so you get good expression of genetic differences. But also, if there's a breakout in one of these herds, people like Kerry won't be able to sell germplasm, right? It's so they have to be kept under high biosecurity. But their offspring, the commercial animals, are typically under some disease pressure, depending on the farm. Course. So then the question is, well, how can you select? All the selection takes place here. How can you select animals at this level that will have progeny that will be more resilient to disease, because you don't get an expression of resilience in the nucleus. Now, you could use genetic markers. You could use genomic selection. Identify genetic markers that are identified at this level, performance under this level, or genomic selection. But again, that requires phenotypes that you're collecting at the commercial level, which is very difficult to do. Or you could have indicator traits, things that you can measure in a high health environment that are good indicator traits to select for disease resilience indirectly. Okay? And that's one of the things that we were after. So to investigate that, to develop those, well, first of all, to understand the genetic basis of disease resilience, but also to identify potential indicator traits, together with several people here in Canada, University of Alberta, University of Saskatchewan, Guelph, Bonnie was also involved, Bonnie and Julie. We got some collaboration from INRA. And also the animal work was all done at CDPQ in Quebec, with funding from Genome Canada, Genome Alberta, Genome Prairie, also ARDAR, and also from USDA. We were able over over basically a 7-year project that this project ran, we were able to leverage the initial investment that was made by Genome Canada. And the idea was to set up an environment with high disease pressure, so the natural disease challenge model, to get a good expression of differences, genetic differences in disease resilience. Okay, using pigs that came from breeding companies that were part of PigGen Canada, the consortium of initially of seven companies, including Genesis. Some of these are not here anymore, but and there have been changes for some of the others. But 7 industry partners that had a nucleus presence in Canada, and that basically gave the industry component of the funding that we were able to get from Genome Canada. And it was very successful, multiple large grants we were able to get through that. And then also PigGen Canada provided the pigs that went into those disease challenges. So this disease challenge was set up at CDPQ, at that test center where they have facilities to also record feed intake and take a lot of blood samples and so on, and very good veterinary oversight. So in their challenge barn, which was here, so just north of the St. Lawrence River, they had a challenge barn that had a late nursery and then the finisher in the same airspace. And they, in 2015, they brought pigs in from neighboring farms that had been where there were diseases present. Okay, so that's how they introduced the diseases, different pathogens, into that barn. And then it was maintained through continuous flow. So every 3 weeks, there was a batch of 60 or 75 just weaned piglets that came from one of the PigGen members and were transported to a quarantine nursery here that was high health. They stayed there for 3 weeks, and then they were introduced into the challenge nursery, and they were exposed to the viruses by horizontal transmission. So in that same barn were the previous 3 week, the pigs that came in 3 weeks earlier that had been exposed to disease. And so that's how the disease pressure was maintained over time, but it still fluctuated. Right, so and there was a lot of things being done, veterinary oversight to make sure that it didn't get out of hand. So a lot of veterinary oversight led by John Harding from the University of Saskatchewan. This is an example of one of the batches. The red ones are the ones that died. A lot of variability, more variability than you would see in a typical, under healthy conditions. The other thing about it is that each batch was from another company. So they provided 7 companies. They provided a batch in a cycle. Okay, so and the aim was not to compare companies, which we couldn't, because each company's pigs were entered as a batch at a different time, different disease pressure. So there was there's no basis for comparison but we were interested in genetic differences between pigs from the same company, within a company. We were looking at genetic variation. So that's the way it was designed. And we got the data, but we don't know which company is which. We don't need to know which company is which. But then the breeding companies, they all have access to their own data. I think about 25% of the pigs died during the challenge study, didn't make it to market. And the reasons for death were, representing the different diseases that are there. A lot of respiratory, a lot of intestinal problems, and some others. And then also the health treatments, mostly treatments with antibiotics. The number of treatments that a pig received, quite a number didn't need any treatment, so they were more resilient, even though they were exposed to diseases, multiple diseases. But then others, they required a lot of treatments. So that's another measurement of resilience that we use. And then it was in a research barn, and so we were able to measure lots of phenotypes: growth, performance, feed intake and behavior, drinking behavior, number of health treatments, mortality, and then also carcass traits for the ones that made it to market. So those are our resilience phenotypes, performance under disease. And then in the quarantine nursery, 3 weeks, a lot of possible samples and measures were taken that could be the basis of an indicator trait. So things we can measure on healthy animals, young healthy animals. So quite a number of immune response traits, including the high immune response that was developed by Bonnie's lab. I'll talk a bit more about it, but also other complete blood count natural antibodies, also omics data, and then one part that I'll talk about in the end is measuring levels of stress hormones in hair, which actually looks very promising. And then they were all genotyped using a SNP panel so that we could all do all the genetic analyses. So in total, about 4,700 pigs. They were all Yorkshire landrace barrows, but from 7 different companies. And this was done over a seven-year period that this data was collected. So the first question that people will ask me is, well, this is not a controlled experiment. It's very messy. The disease pressure fluctuates. So how can you ever get good data out of this? Well, a good test for whether you have reliable data is to evaluate whether the traits are heritable. Okay, but if the trait is all noise, it's not going to be heritable either, right? So we estimate. So Austin Putz and Mark Cheng, 2 graduate students at the time, PhD students, they estimated heritabilities for the performance traits under disease and for the health treatments. We also had subjective health scores, and they were all. The performance traits were as heritable as you would get the heritability you would get in a nucleus herd. Doesn't mean it's the same trait, but it was quite heritable, so that it does provide reliable data that has a genetic signal. And then these treatment rates and mortality rates lower heritabilities, but higher than what you would typically see from commercial data. Okay? Partially because the incidence is higher, but reasonable heritability for those clinical traits also. When we look at, so we also had on several of these the pure lines that produced these Yorkshire landrace barrows, we had access to their nucleus data. So we were able to estimate the genetic correlation between the performance of the crossbreds in this challenge to the performance of their purebred parental lines in a clean environment. And those genetic correlations were pretty low, 5 different lines. If you look at average daily gain in the finisher, the highest is 0.6, but as low as 0.1 genetic correlation. So indicating that although the heritability of average daily gain in this environment and in the nucleus environment is similar, they're genetically very different traits. Okay, and as you would expect, and the same for feed intake. And then Mark Cheng also did a GWAS, and this is just an example growth rate in the challenge nursery and he finds a big effect on the major histocompatibility complex. And if you look at GWAS for growth rate in nucleus data, it's not there. Okay? And it makes sense that it's here, right? But that explains part of the reason why the genetic correlation is different, because there are different genes involved to maintain growth rate under a severe disease challenge. And then also, he also found that region, MHC, also explained substantial variation for mortality, number of treatments, health score, and then also average daily gain in the finisher. And explained a big part of the genetic correlation of those traits with growth rate in the challenge nursery. the genetic correlations for the MHC region were in absolute value quite a bit larger than the genome-wide correlations between the trait on their own. So indicating that that MHC region is important for growth rate under challenge, but also mortality, number of treatments, and so on. So, of course, with MHC, you don't know what you want to select for because you do want to maintain some diversity there. We also went back to the GBP5 gene that we had found to be a major gene for resistance to PRRS. And that was also significant for a number of treatments under this challenge where we don't just have PRRS, but we have lots of other diseases also, whereas the initial finding of GBP5 was on an experiment where they were infected just with PRRS. Okay, so showing that region is also important when you have multiple pathogens present. And we did also look at CD163. So CD163 is the gene that was knocked out to create resistant pigs for PRRS. There are SNPs in that region, and we did find that there were some associations. They weren't strong, and they were also not very consistent between companies. So, but there is some natural. Apparently, it did indicate that there's some natural variation in or around the CD163 gene, but no variants that make them completely resistant. Okay, so now let's talk about these indicator traits. Now, for a trait that is measured here to be a good trait to select for here to improve resilience at this level, it has to be heritable. It has to be measurable on healthy pigs. It has to be heritable, and then of course it has to be genetically correlated with disease resilience. So that's the type of indicator traits that we looked at. The first one. Austin looked at feed intake patterns under disease. So we had daily feed intake on these pigs that were in the challenge, and the red dots here indicate when the pigs got treatments. And of course, what he saw is that they coincide with drops in feed intake, as you would expect. And so he used that to try to use this feed intake data as an indicator trait. So he looked at day-to-day variation in feed intake. So he put a regression line through. This is data of one pig, a regression line, and then the residual variance from that regression line he called day-to-day variation in feed intake. And the idea was that if pigs have fluctuate more in feed intake from day to day, they may be less resilient because they're more affected by the diseases that come along. And then he also identified the percent off-feed days based on a criterion. And then he and also Mark Chang, they looked at the heritability of that. Not that high, but he also looked at it based on, instead of feed intake, having on the y-axis how long they were in the feeder per day. Okay, and that was more heritable. And then this shows the genetic correlations of these traits with the treatment rate, and they were all positive. So this is for for the different in the different phases and different treatment of the data, but generally indicating that pigs that had higher variability or higher off-feed days, that they tended to have higher genetics for susceptible to more treatment and higher mortality. Okay, so indicating that these may be indicator traits that could be used to select for disease resilience. But of course, this is under disease. We subsequently have also asked the question, well, if you look at day-to-day variation in a nucleus environment on purebreds, the breeding companies, they collect a lot of feed intake data. Is that heritable? And we also found that to be similarly heritable, but it wasn't highly genetically correlated with disease resilience. So that wasn't a very clear signal that feed intake, day-to-day variation feed intake in the nucleus could be used as an indicator trait, just like for resilience. So then we also looked at different immune response assays. I mentioned some of these already. And then again, we measured them on a number of animals in the quarantine nursery, and then estimated heritabilities and then genetic correlations with the resilience traits, the resilience phenotypes. And we published a lot. I won't go through all of those, but the one that I want to highlight, and that but the most promising, is the high immune response technology that Bonnie and her lab, Laurie, was involved in developing that for dairy cattle, and I was actually still here when that was developed, and so that of course is so the two types, the two arms of the immune response, cell mediated and antibody mediated. I'm not going to spend a lot of time on here, but this is from work from Laurie from your master's thesis, and showing that cows that had higher immune response based on these two tests had less disease. Okay, and that is now has been for quite a number of years already been used by CMAX as an immunity plus to identify bulls that sire daughters that are more resistant, a better immune response. Okay, it's been very successful. So then, when we started this project, people from PigGen Canada and Mike Dick from Alberta approached Bonnie, and then Julie came on board to implement this for pigs. It was actually initially developed for pigs, right? And then applied to dairy cattle, but here we convinced Bonnie and Julie to go back to pigs and try this again in pigs under these circumstances. So that the measurement was done in the quarantine nursery, right? So the antibody-mediated and cell-mediated response was measured, and then we estimated heritability. So Mark Cheng, my PhD student, did the genetic analysis. I found them to be fairly heritable, similar to what heritability is in dairy cattle. The antibody-mediated had a higher litter effect. And then and then also the genetic correlation between the two arms of the immune system were low positively correlated, or the standard error was high, and that's closer to zero in dairy cattle, I believe, right? Yeah. And then, of course, we were interested, well, is it a good genetic indicator for disease resilience? So we estimated genetic correlations of these traits with growth rate under challenge, the number of treatments, and mortality. And there we found, a surprising result. First of all, the antibody-mediated immune response, showed genetic correlations that were in the expected direction. In other words, that pigs that had better antibody-mediated immune response genetically were more resilient. They had higher growth rate under disease, especially when the major challenge happened. They required fewer treatments and had lower mortality. But CMIR, the cell-mediated response, tended to go the opposite direction. And we're still not entirely clear why that is. It may be because of the nature of these infections that are very bacterial oriented. We forget.
I guess the treatment data is very
Okay. Yeah, that's right. Yeah, because we're looking more. Yeah. So most of the treatment data is what treatment was given, and a lot of that is antibody related. Okay. So there may be some biases there, and we're not suggesting that you should select for lower cell mediated and higher AMER, because that kind of pulls the immune system out of balance. Probably still want to select for both of them to be higher. Okay. But additional research clearly is needed. But it showed some encouraging genetic correlations, especially for AMER. Then we also did a lot of omics data. Actually, so this was done on over almost 2,300 pigs, 2,300 of the 4,500 pigs. We had AMER on. For the omics, we had fewer individuals, about 900 for most of them. Although for transcriptomics, we now have 2,500 of these pigs have blood transcriptomics. So all of these omics data is all based on blood, except the fecal microbiome. But basically, again, those are samples, blood or feces that were taken in when they were still healthy. There's several papers here that we have. Again, some encouraging results, but the numbers are the number of animals that we have these data on is too limited to draw strong conclusions. But there's some promising results on. So my postdoc, Kyu-Sung Lim, who's now back in South Korea in the faculty position, he looked at the gene expression on young healthy pigs as a source of biomarker. So blood was taken in the quarantine nursery, and then we did 3' RNA-seq. So to keep the cost down, rather than doing full RNA-seq, this only sequences the 3' end of every transcript. Okay, so it's a lot cheaper, and we were able to get to, actually we're up to 2,500 pigs of these pigs that now have gene expression data. And he's published 2 papers on that, including some genetic analysis, and we're doing some additional genetic analyses now. Huge data and show some interesting results, but it's a database that, we talked with Ray this afternoon about, one of the genes that I'll talk about in a second. We can now also look in this database, well, are there differences in gene expression between pigs that have different genotypes for QTL, for specific QTL, and look at possibly networks of genes. The last part I'm going to talk about is stress hormones in hair. So we know that stress can be invoked by lots of things, disease, but work, whatever, late nights, fear, and everything. And it, of course, it acts through the HPA axis, and it produces cortisol in blood, DHEA, which is sort of an anti-glucocorticoid. Also cortisone, which is more or less the inactive form of the storage form of cortisol, and also the sulfided form of DHEA, which is sort of the storage form of DHEA. So these 4 hormones we measured in hair, because as this is as a result of stress, higher levels of these hormones are produced in blood. That level, or that gets incorporated in hair as it grows. Okay, and so the idea is that if you cut the hair and then analyze the level of those hormones in this hair, you're measuring the total or the cumulative or the entire the level of those hormones in blood during the time that this hair was grown. Okay, so it's not an acute measurement, but it's a measurement that captures weeks or months. Okay? For those of you who have long hair, you can look at the tip of your hair and look at how much stress you were under when that hair was grown versus other sections of your hair. We didn't do that. We actually we cut the hair, the same patch on the back. We cut it just before they went into the challenge. So then we're looking at hair that was grown when the pigs were not challenged by major diseases, but they were under all kinds of other stresses. they're being born, they have to get used to drinking milk and finding the teats and so on. They interact with their litter mates, then they're weaned, then they're transported from one side of Canada to the next. They are mixed again, they're a new diet. Okay, so lots of stresses, very stressful time that the time since birth until several weeks after weaning. And then that same patch, the regrowth, was shaved again, I think 40 days after they entered the challenge nursery. So this is measuring more response to infectious stressors, where this is measuring more response to non-infectious stressors. Okay? And we had different numbers because we lose some animals that don't make it to this point. And not huge numbers, but enough to be able to get some interesting results. So first of all, we looked at heritabilities. So the blue lines are from the first haircut, the red is from the second haircut. You can see the cortisol is fairly heritable for both haircuts. Cortisone only in the second, and then these alternated. Not entirely sure the reason, but overall we got some good heritability. So as far as an indicator traits, there's something there. We also looked at the phenotypic and the genetic correlations between, for example, cortisol in that first haircut and the second haircut. And that was significantly correlated, 0.5, not huge, but so genetically, the cortisol we measure in those two hair samples under those two very different stressors, although there's also a difference in age. They were moderately correlated genetically. Less so for cortisone and also, well, these have large standard errors because one of the two was not heritable. Okay, so especially cortisol, and that turns out to be our probably our most useful hormone to measure. And all this work was done. The assays were all done by Yolande Seddon, who is at the University of Saskatchewan, and involves a lot of work because it involves cleaning the hair from contamination. They actually cut the split ends because you may have more erosion of the hormones because they're more exposed to air, and then grinding it and then doing ELISAs to measure those hormones. So a lot of work. So really want to give credit to them as well as the people that did all the animal work. We did a GWAS and we found a major gene. Biggest QTL I've ever found. We thought that with the GBP found a big effect. Explain about 15% of genetic variance. This explains over 40% of genetic variance. And it's the same window for cortisol level in the first haircut and the second haircut. There's something else here that, and then this is very close, or it includes the glucocorticoid receptor gene, which is the receptor that binds cortisol and cortisone. So biologically, that makes a lot of sense. And also previous work out of Germany, actually, found for cortisol in serum after acute stress, they found a mutation in that gene that was associated with level of cortisol in serum after that's acute stress. We did not have that mutation in our panel, but we imputed it and didn't find an effect. So we think it's a different mutation, but we're hoping to have funding to have a collaboration with Ray to dig into this and to validate these effects and to hopefully find a causative mutation. But a huge effect. And this, I remember I was talking about the genetic correlation between cortisol in the two haircuts, about 0.52. If we adjust for this major gene, it's almost zero. So this genetic correlation is almost all explained by this major gene. Okay. And this is the effect of that. So the minor allele, the allele that decreases cortisol, lower stress response, it's fairly low frequency, and actually in one company it wasn't present. Okay? So we don't have pigs that are homozygous for the minor allele, or very few. But so here we're comparing the ones that are homozygous for the allele that increases cortisol to the heterozygotes, and the heterozygotes have 30% lower cortisol level at both haircuts compared to the homozygous. So that's a huge effect. Also a big effect on cortisone, less on the other two hormones, but a huge effect. And then we are interested in, well, is this major gene, is it associated with disease resilience? So this shows, there were actually 2 SNPs that showed very large effects in that window. And let's see. So here, and this is the effect again of the minor allele, so the effect of the allele that decreases cortisol. Those pigs that genetically had, the heterozygotes had genetically, well, there was some evidence of a higher growth rate before they went into the challenge, not much difference, no significance during the challenge, but there was a significant difference in feed intake. So the pigs that had the minor allele that reduces cortisol genetically had higher feed intake and under challenge. And that's what you need, right? You need pigs that keep eating, right? And so that's we view that as an indicator of resilience. There wasn't much effect on feed conversion ratio. Mortality. So there were significant effects on mortality with pigs that had the allele that reduces cortisol had lower mortality. Okay, they had, well, if you look at this one in the finisher, they had higher health scores in the finisher, better health scores, and there wasn't much of an effect, significant effect on health treatments. But across the board, this did indicate that this major gene, the allele that reduces cortisol, is beneficial for disease resilience. So it could be used to select for disease resilience. Another interesting finding was the effect on carcass traits. So the allele that decreases cortisol, those pigs had poorer meat quality. They had lower lean yield, lower dressing percentage, and the tendency for higher back fat. Now, when we saw that, that reminded me of the stress gene, the halothane gene. Some of you are too young to remember that maybe, but the stress gene was identified in the 70s, I think. The causal gene was found by at the University of Toronto, is associated, it's the ryanodine receptor gene involved in calcium transport. But also the pigs that have that gene, or they have the stress gene, the stress allele, they when you anesthetize them with halothane, they stiffen up, okay? And they also are more stressed, and it also creates poorer meat quality, because during transportation they're more stressed, and you get all the negative effects of that. They have poorer meat quality. Well, that's exactly what we see for this minor allele, right? So the ones that are stressed, that have the stress gene, actually have better meat quality. That's the way it went. Because that's the reason why the frequency of the halothane gene became quite high, because breeders started to select for lower back fat and higher leanness, and they indirectly increased the frequency of the stress gene. And here we see the same effect. So if you look at the major allele that increases stress response has higher meat quality. Okay, so the same effect, different mechanism, we think, but the same effect, the same kind of relationship. So then we also looked at, well, what about What about the genetic correlation? So the previous was just looking at that one gene, but what about the genetic correlation of hormone levels with performance and disease resilience trait? And we did those analyses both with and without the major gene fitted as a fixed effect, and these correlations actually didn't change much. So this is the level of hair hormone in hair cut or grown under disease. And it's a lot of data here, but let me point out some things. So here, the pigs that had higher level of especially cortisol, if you focus on the blue ones, of cortisol, and to some degree also cortisone, they tended to have a higher level of cortisol, lower growth rate. Okay, so that's in the expected direction, and the same with feed intake. Okay, so indicating that if you select pigs that have lower levels of cortisol in hair that's grown under challenge, you expect them to have a better growth rate under challenge and better feed intake under challenge. Sorry, that the ones that have lower cortisol. FCR, there was some indication that it was to higher, lower feed conversion ratio. Mortality, so that what went in also in the expected direction for cortisol, that the ones that have higher cortisol have higher mortality, more stressed. And then for health scores, it tended to be negative in the expected direction, that the ones that had high cortisol had poorer health scores. And then for treatment, not significant, but it went into the expected direction. And then also for carcass traits, it went in the direction that we expected, that the ones that are more responsive to stress have better leanness, same as the stress gene. So that was for hair measured grown under the challenge. This is the ideal hair for an indicator trait, hair that is obtained from pigs that are still healthy. And there we didn't find much of a genetic correlation. There's some signals there, but not very consistent. And so that doesn't look like a very good indicator trait. But the marker we identified from there is, right? So you can still select for the marker. Okay, so conclusions. So we do think that disease resilience is a useful target to improve response to multiple pathogens. But of course, it's difficult to evaluate in the nucleus environment. It doesn't mean that it's not useful to go after resistance genes for specific diseases like PRRS or African swine fever. I think that's still important, but getting gene out of the pigs that are resistant to all the pathogens they're exposed to is going to be very difficult to achieve, and so there's still always going to be a need for pigs that are may get infected, but are better able to deal with those infections. Okay, so yeah, this polymicrobial natural disease challenge model seemed to be pretty effective in generating good data. We can use that for genomic prediction or developing genetic indicator traits. So we talk about day-to-day variation in feed intake, several immune response assay, blood omics data, and then the one that, in addition to HIR, the one that's most promising is the levels of stress hormone in hair. So they were moderately heritable, especially cortisol, and they were genetically correlated between hair grown under disease and on healthy animals. But most of that genetic correlation was driven by that major gene near the cortical receptor that explained over 40% of the genetic variants. And that can be used directly for selecting pigs that are more resilient without having to expose them to disease. Okay? It would be good if we can validate this and find the cause of mutation and understand more about the mechanisms, but that can be used already. And then also measuring stress hormones, especially cortisol, in hair of grown under disease. And so that could be done by collecting shaving hair from pigs that are in commercial herds that have disease outbreaks. It could be a source of phenotypes to evaluate, and then through genomics bring that back into the nucleus. We also did look at the back test. I didn't talk about that, but that's basically putting the piglets, nursery piglets, on their back, and then measuring how they behave, how often, how intense they struggle and scream. Okay? And they were heritable, and there was also some indication that they were that the pigs that behaved better under that back test, they were genetically more resilient, but needs to be validated. So these are a bunch of the students that were involved in postdocs, but there were a lot of people at Guelph from here, at University of Saskatchewan, University of Alberta, at CDPQ, or the staff there. They did an amazing job, led by Frédéric Fortin. And then, of course, the breeding companies that provided not only the pigs, but also funding. We also got some funding from the National Pork Board and Iowa Pork Producers Association, but then major funding from Genome Canada, Genome Alberta, Genome Prairie, and also USDA. So with that, final thing, registration is open. The World Congress, July next year. All of you, the abstracts are due in couple of weeks. So hopefully, and then the 4-page papers will be due January 1. So I'm one of the organizers of this Congress. You can go to the website, and it has all the information for submitting abstracts and part of the program. But hopefully, many of you, I hope to see many of you there with presentations, and beautiful venue, conference center here right on the lake. It was designed by a very famous architecture, Frank Lloyd Wright, in a beautiful city, Madison. And yeah, we hope to make it a good event. So we call this the Olympics, every 4 years, held every 4 years, the Olympics or the World Cup of animal breeding. So hope you can all make it. So thank you. Happy to answer any questions if there are any. And if there's
Thank
time.
you very much, Dr. Dekkers, for your presentation. I would like now to open the floor to any questions, either from those here in person or from those joining us online. Please, Ricarda.
How many did you have multiple generations of stress data that you had data on the stress for multiple generations? So maybe a sow and then offspring, or did you have one generation of?
Well, it was over a 7-year period, right? So in total we had, I think we had 65 batches spread across seven companies. We had 9 to 10 batches per company, and so yeah, they would involve multiple generations.
Then I have a follow-up question.
Yeah.
Did you look for epigenetic effects over the generations?
No, we have not looked for epigenetic, and of course the parents were not challenged, right? The parents are in the nucleus, right? So and the ones that are challenged are terminal. So yeah. Sorry.
About the G loss. Under affected, there was a marker right at chromosome 14. Do you know what gene that was?
Not the top of my head. I think this has been published. This paper has been published, so it should be there. Should be in there, but. Yeah. So your question is about.
Yeah, that marker.
About
right
this
there.
marker here, yeah. I forget. I mean, it's not a huge effect. It explains there's a jump in the axis
Oh, I
here.
see now. I see
So
the line. Okay.
yeah, less than 1.5.
Right.
It's not a huge effect.
Okay, sorry. From this angle, I didn't see the line.
but I see it now. But I have another question.
Yeah.
So you wouldn't have expected, less stress to result in weaker carcass traits in the hair. So how do you explain that? And because we would think that less stress would be, more calming, more intake of feed, and better gain carcass traits as a result. But you saw that the minor allele, it was the minor allele.
The minor
that-
allele reduces cortisol.
Reduces cortisol, unfortunately affects carcass trait
Yeah.
negatively.
Yeah.
So
I don't have a good explanation for that.
it's carcass traits, not growth, right?
Yeah.
Yeah, it was just the yeah. So anyhow, just how do you resolve that, or how do you reconcile that?
Well, we have to know more about the mechanism, right? And so you're talking about, let's see.
That upside down pig, the halothane pig. What was the mechanism there again? That was calcium?
Yeah,
So
calcium.
maybe it's something about, calcium getting into that carcass.
Well, yeah, but that was the ryanodine. That's the ryanodine receptor, which directly involved in calcium transport. But
Positively? Like, because calcium, because almost every cell signal needs calcium.
okay.
Right? So I'm just trying to figure out what is it about and what particular carcass traits were benefiting. Like, is it loin eye area or like does it have to do with the size of the muscle? Like, if we could figure out what it actually is doing, we could maybe figure out what the calcium is triggering.
Yeah, so I mean, this is the extent of data we have. So we have dressing percentage, loin depth,
Okay, so again, size, right?
and well, loin depth, and this is loin yield and loin depth,
Okay,
and then back fat went the other way, other direction.
going on there. So it's the cell signal of it.
Yeah.
Sure, you'd think.
I don't know, Ray, you've worked a lot with the glucocorticoid receptor. Anything connected with calcium transport?
I'm not too familiar with the calcium, just like Bonnie was saying, calcium just connect with so many things.
Everything.
Almost you can say anything is correct, right? So
Yeah.
I cannot, on top of my mind, to have a direct correlation of calcium and stress, because it's just too complicated. For the stress part, this is something, the more I read and the more confused I get. Because there are two kinds of stress. One is called eustress. You got to be stressed actually good for you. Like people, you run on treadmills, it's good. But if a lion chase you through the woods or whatever, that's a wrong stress. You're also running. Like to what extent running is good, run and becomes bad.
Yeah.
This is something like I couldn't figure it out. So therefore, in this study, I think it's very interesting. You see a correlation, but if you want to tease out which part of stress is beneficial, which part of stress is bad, I cannot say all stress is bad.
No.
And also here, I think at the conference, I talked to Jack and also Yulan, and we had a good chat. Is that here you measure the chronic stress, right? Because the hair, we actually have done something to measure the cellulite stress from the pigs. So the acute stress and chronic stress, not directly linked to eustress or bad stress or stress, but also they're different. So I'm just thinking that the halothane gene, when there's a heat shock and the pig died, I would say that's acute stress. So therefore may not link to the hair hormone.
That's true. Yeah.
So therefore, when you look at the mortality, I would look at the same thing. Why mortality and carcass trait is like that? Maybe it's different kind of stress.
I have a question to that. Would stress be kind of like G-forces? Because if you look at G-forces up to 3 or 4, a human has fun, right? In a roller coaster, if you encounter a G-force of 7, you're knocked out, right? You see all the.
That's a bad stress for sure.
Yeah, is it like a threshold maybe?
I don't know. I really don't know. Like I think it's very vaguely defined. Like what is a eustress?
The other person to talk to is Neil Carroll.
Mm.
He was gonna be here, but he had to teach, because he's like a stress expert and he's been, working a lot. But one thing he always reminds me is cortisol has all kinds of beneficial effects. We just think of cortisol as the bad guy. That's what gets you up in the morning,
Yep.
right? That's what's the last thing in lung development before,
Yeah.
a baby's born. So, but what I like about this is you are getting at the chronic stress in that hair over time.
Mhm.
So you're pretty well assured you're in the bad area, I think. So it's kind of I think it's a really interesting thing. And the fact that your SNP is right probably near the receptor
Mhm.
makes complete sense. So
Yeah.
my guess would be go in and it's probably a receptor because cortisol doesn't work much without a receptor.
That's right.
So I don't know, it's pretty interesting.
And that's what in the German study, what they found for acute stress, the causal mutation in that gene or near that gene, it reduced the affinity of cortisol to the receptor.
Yep.
Yeah.
There's something going on. It's cool.
Would
From a breeding company perspective, is it something that you would suggest breeding companies start selecting for or against?
That marker?
Yeah.
Or
do we need more studies?
I think we need validation, and that's what we're hoping to be able to do. We just submitted with Ray and Yolande and Mike Dicker LOI to Ardar. So hopefully we'll be able to do that validation. Yeah, because they were imputed. Yeah, go ahead.
I have a question online.
Okay.
Is the bed?
Yes, I have a question.
Yeah.
Yes, I was always told by Larry and people that you guys never got excited about major genes because went to fixation so quickly. So my question for you, and this is maybe a bit naive, is how come you have this gene of major effects still in your population? It sounds like a balancing selection to me, like, sickle cell anemia and stuff. Could that be true? Over.
Well, I think the reason why we had the same question with the PERS major gene for PERS, why is it still in the population? Is because there is no selection under disease challenge. Selection is in a healthy environment, right? So there's no selection pressure for selecting animals that are more resilient. So that's why I think these alleles are still in the population.
That's cool. Thank you.
Yeah, good question.
So I have one more for you, Jack. So with all of these traits, have you done or do you know that has principal component analysis been looked at to potentially look for any confounding factors? Has it been looked at, or is there an interest in looking at it to try and explain some of the things that we might not expect to have seen?
Yeah, so one of my PhD students looked at all these different immune response traits, and he has started to look at genetic correlations among them, and he's also starting to look at principal component analysis. So because there's a lot of I wouldn't say confounding, but they measure some aspects of the immune response.
So it's already covered.
Well, natural antibodies, for example, and antibody levels, and these phagocytases. I mean, they're all part of the immune system, right? And that's all connected.
It's all connected on pathways, multiple pathways. Multi-genetic control.
Yeah. So I think the idea is that if we look at all of these together, we may be able to draw more out. Yeah.
Sure.
Yeah.
Just a comment on the PERS situation where you've got a major gene, which happens to be the receptor for the virus. So when you take that approach, you often will get a short-term patch, but those viruses will never, ever stop. because they are going, and that's why you're probably seeing some association of that receptor with some other pathogens, because almost never is there one single receptor.
Mhm.
So when one is no longer beneficial, that virus, that especially if it's a highly mutating virus, it'll just start and swing to different receptor.
Yeah.
So that's the trick. You'll get an effect in the short term, but I'm not sure how long it will last.
That is definitely one of the concerns. But then, I talked to Ray about this over lunch. If that were like the PRRS virus, there must be some severe blocks because the PRRS virus, as far as we know, hasn't jumped to humans. Even though the farmers are inundated, well, when they work in infected herds, they are exposed to it.
Yeah.
Day in, day out.
But then you just look at what's the receptor in humans. So if it's substantially different, right, it won't jump species. That's much more difficult
Okay.
than within a species doing a modification and that virus just saying right within the species I'm very adapted to, because it is adapted. It's not like flu, which is adapted across multiple species.
Yeah.
That one's adapted there, and it could, but it's not guaranteed. You don't know what it's going to do. But my guess would be you'd get an effect. I don't know how long it would last.
Yeah, yeah. Well, yeah, and I think that we have to keep monitoring, but also for these approaches. Also, if you select on an indicator trait, you have to keep monitoring, make sure that the association, that genetic correlation is still there, because it can change.
We have time for one last question. If no one has, I have a question. Okay, so you said that you're going to look genetic correlations between the immune traits that you measured. Is there any plan, or did you look the correlation between these immune traits and the cortisol level?
No, we have not done that yet, but yeah, we have not done that yet. That's a good suggestion. Also.
Because we know high cortisol chronic is going to decrease immune response. That's well established. Especially that chronic, and that chronic stress is also in humans associated with things like cancer, these chronic disease problems. So that's a really great thing to have a look at.
Yeah. Thanks.
Okay, thank you, Dr.
Okay, thank you. Thank you.
This has been a great discussion. Thank you all for coming today, and see you all tomorrow.
Thank you. No
No alarm.
it was a false.